So our next speaker is Dr. Sreeja, Assistant Professor Christ. Over to you, ma'am. Thank you.
Yes, sir. Thank you, sir.
Yeah.
So very good afternoon to one and all present here. Thank you for the opportunity given for presenting in this conference, and my apologies for not being there. So what I'm going to talk today is, the title is Anti-counterfeiting method for drugs using synthetic DNA cryptography. As you can see, there are many keywords like anti-counterfeiting, synthetic DNA, and then DNA cryptography also plays a role, which I'll be discussing on the go. So what I'm going to discuss, just an outline. So a introduction about how this topic has been framed, a little bit background about all the keywords which I had specified, the related work, little bit analysis part, why we are going for this particular research, POC followed by analysis, conclusion. So as I had specified earlier, counterfeiting is one of the keyword which actually made this research most prominent because of the serious issues which are happening right now. Counterfeiting is something which was existing, still is, and actually it has a huge impact right now because of COVID-19. The COVID-19 connect is that, as you all know, the medical equipments and other drugs all are actually right now being sold online and offline. Right now it has increased. So the data says the COVID-19 pandemic, even the sanitizers what we use, like the face mask, all those things are counterfeited. So when I say counterfeited products, what are these type of products indicates what? The products which are not genuine, or the products which, the authenticity we are not able to claim, because we do not know the source of the products. That's what we call as counterfeit products, though there are many definitions, including the definitions given by World Health Organization. So why we are talking about this topic and why it has relevance? Because counterfeit products are a persistent and direct threat, definitely to economy, business, sometimes including the national security when we look at war weapons and so on. It is affecting all of us. But why this is more relevant when it comes to health? Because it is directly affecting life. There are many cases which has been reported. The counterfeit products, that is especially drugs, are leading to mortality. The data had not included much here, but yeah, there are references given. Now, what are commonly counterfeited products? So these are few of the products which you can see straight from water filters to so on, electronics. But what we are focusing is mainly drugs, considering the fact India is actually, if you look at the third position, it is maintaining the entire supply chain with respect to drugs also. These are the incidence regions of the world, which shows how important this topic has to be addressed, especially the research community has to look into this particular topic. This has been provided by Pharmaceutical Security Institute, which is a non-profit organization. The sources given here, it's just the data to show how it is prominent, even after we are having many techniques to avoid this. So as this is one of the definition, just to indicate, it is a medicine, either the ingredient is not proper, or it could be the mislabeled one, or maybe the ratio in the active ingredients might be an issue, or maybe the packaging, whatever it is, the fake packaging is also something which we will consider. So it all talks about the product which is not genuine. It can be the packaging we are looking at, or even the drug ingredients, that is the active ingredients what we are looking at, or maybe the assay of the product when it comes to drugs. Now, what is anti-counterfeiting? Any technique which we are going to use to prevent counterfeiting is known as anti-counterfeiting technique. Now, how many techniques we do have? The research says we do have 3 different types. These are the categories which I had consolidated, including overt, covert, and then forensic features. When I say overt features, something which we had been used, like holograms is one of the example. And when we come to covert features, it can be even the invisible ink, or maybe the watermarks what we use. There are many different categories what we use. Then let us go for forensic feature, which indicate a few of them, like chemical reagents what we use, and then comes another category of DNA taggings. So though we say forensic feature, the literature says the forensic features is most prominent in terms of security, but least in terms of usability. So that's where the need for this particular topic comes into picture. So though we have the forensic features, as I mentioned, it's all in vitro. Like we have to use the reagent, maybe the dedicated devices we need. So how do we ensure, as a user, if I want to ensure the product, whatever I had received, right, is authentic or not? That is authenticity. How do I claim it is genuine or not? Something of high relevance right now. So there comes the importance of the draft which has been released by WHO. So what do they say about anti-counterfeit technology. They list many techniques which we can use, including the forensic markers, chemical taggants, whatever we want to use, biological taggants, and especially comes DNA tags. So as you can see, DNA taggants here, they say it is secure. So there are various factors which we have to consider. As we all say security, definitely security comes. If you look at high-end security, maybe usability is something we can think of, but then sometimes what happens, if it is a product which I'm considering is genuine, but if it takes longer time to ensure the authenticity of the product, how do I ensure usability? This is where, as research community, we can contribute with our information security techniques for the products which we are using, especially in case of drugs. So how do we ensure this anti-counterfeiting is achieved, or maybe counterfeiting is prevented? That's where all our encryption techniques, including PKI, is also used when we come for anti-counterfeiting, especially with respect to RFID is what so far it has been used, especially in supply chain and all. So here comes a new technique. When I say new technique, what is new here? I'll explain on the go. Now comes the DNA, that is the DNA keyword which I had specified. We all know DNA, right? When it was discovered, the structure, the helical structure, double helix structure, it was discovered in 1953. We all fascinated, and that also laid the foundation for modern molecular biology. But what has fascinated as people working in information security more is the usage of DNA computation and the related principle in terms of security, especially in when 1994, Dr. Adelman proposed how DNA can be used, DNA computation can be used for security. He solved the problem by finding the shortest path, that is in vitro. Later on, it was developed and modified to various levels. So DNA cryptography is something which has been used right now for achieving confidentiality, authentication, signature, including e-signatures, is something which we can see where it is widely used. So previous talks was actually relating to homomorphic encryption and how when quantum computers comes into picture, what are the vulnerabilities of the existing protocols and so on. Right? This is where actually importance of DNA computing also we can think of, because it is not the conventional way of computation, but we are relying on the principles of DNA. That is, say there are various principles which we can look at. One of the example could be central dogma of molecular biology. We call it as CDMB, where the transcription and translation process can be considered for encryption. So this is where the relevance of using DNA computing for information security comes into picture. This shows the basic structure which has been adopted from nature. The source is nature. As you can see, GCAT. So this is nothing but a polymer, and we can consist, like each is monomer. It is made of monomer units, where we have a little bit of biology, where the bases, nitrogenous base we have. So we will be looking more at AGCT and how this works with respect to encryption, how we are using this basis, and definitely the database which has been created by NCBI and EMBL helps us to collect the data which we can use for encryption. But going further, we can also see, instead of taking existing data related, that is the database is what we have as a part of Human Genome Project is one source. Secondly, we can also see there are various tools which is helping us to create DNA sequences. One of the tool is Gene Designer, which I had used for this particular research, where I can create my own random DNA sequences. So the ethical, whatever the concepts which we connect with respect to ethical and other issues can be considered. Now, what is synthetic DNA? I did mention about DNA. I did mention about what is DNA. Cryptography is about using the DNA principles. Let us talk about what is synthetic DNA. There are various perspectives when we say synthetic biology, or maybe synthetic engineering is a concept we can consider here. It has different perspective. When we look at a point of biologist, it is different. When we look at a point from chemist, it has different definition. So we are considering here with respect to in terms of biology. So synthetic biology, as you can see, something which does not exist in nature. Right? It can be maybe biological parts, or maybe any devices or systems which we are considering, which do not exist in nature, and then I redesign. Right? So this is where actually synthetic concept comes into picture. Synthetic biology comes into picture. As you can see here, ATGC, the basis which I had specified before, the image has been adapted from Synthrax. As you can see, ATGC is given here. Now, what comes next in this particular picture is You can see XY, which are the two bases which has been added. Similarly, there is a combination here, YX. So it gives 3 base pairs. What we can see here, AT, like basically these two are always connected, ANT and then CNG. But here what you can see, apart from ANT and CNG, there is also X and Y. So that's where the 3 base pairs comes in. So basically, we are creating synthetic DNA, which does not exist in nature. So there comes the importance of randomness. We always talk about randomness, whether we are developing a unique code or maybe, when we encode something, how random is the code which you are generating in terms of security, right? This is where the importance of using synthetic DNA comes into picture. So that's about synthetic DNA. There are various works which are connected to synthetic DNA, and research is still ongoing. So you can see DNA-based authentication, when we talk about, it's an area which needs definitely more exploration, but it's an emerging paradigm as it uses biocomputing plus the conventional cryptography techniques. Now you can see, as I mentioned, DNA taggings, it ensures authentication, but there are certain criteria which we have to look into. Sometimes it can be drawbacks as well, as we have to rely on reagents, and then know how to use a dedicated device when I have to check whether the tag in connector is secure or not. But as you can see here, there is a technique which has been proposed, which is a patented technique, DNA lock, which uses PCR. PCR is a polymerase chain reaction. PCR amplification is used here, and by Carter Bancroft. So this is one of the technique which has been already used. Apart from this, there are various DNA-based user authentication protocols which has been already implemented and used. And then, as I mentioned, one for user authentication. So here, basically, we are talking with respect to product authentication. As I mentioned, the drugs, right? Drugs is what I had considered. It can be used for any product, but drugs is considered because of the relevance it has. Now, as you can see, though we have many techniques, now you might be thinking, we do already we have RFIDs, we can connect RFID with PKI and so on, right? This is another paper which shows, though we have anti-counterfeiting techniques, right? These are duplicated, including RFID, whereas DNA preempts counterfeiting. This is what actually motivated me to think about how we can use DNA and how it can be applied to resist counterfeiting. So the unique and uncopyable nature, when I say uncopyable, we always talk with respect to reverse engineering, right? We always say we are good at reverse engineering. If I have a code, right, how I'm going to regenerate the code? Is it possible, or maybe I have to decode it to get the original code? So this is where the importance of DNA comes into picture. I cannot copy it. Even if I copy it, how I'm going to retrieve the original data? That's the challenge. Now, this is the analysis part which I have done to continue with the research. So the increase in fake products, because right now we all know health is wealth. And then the usability, the most important is with respect to brand integrity. Once I took a medicine which I found out it's a fake product, then it's affecting brand integrity as well, right? And also, though we have many devices available, maybe some labels, as I mentioned, all the overt, and then forensic markers, even if we consider usability is an issue, plus the additional cost we have to look for. Even for 10 rupees medicine, I cannot go for, right, using DNA taggings, which need so much of cost. So whatever we are receiving right now, whether it is the drugs or maybe the products or all the security seals, whatever we are receiving, are they intact? Is something we are always looking for, right? Whether I can verify the drug, whatever I had received, is proper or not. If yes, as a layman, how do I ensure the usability to check that? That's where a scheme, a proposed system is what I'm showing right now. This is actually used synthetic DNA to come up with a scheme which can be used without knowing, right, okay, what that code indicates, but definitely it does shows it's the product is genuine. Should say this is secure, right? I should say this is secure plus usable. That's where the proposed system has come into picture here. So I had generated random DNA using gene designer tool. They always connect together by hydrogen bonds. It's known as Watson-Crick complementary rule. That's the phase 2. This entire system has 5 phases. Phase 1 is generating the random sequences, and phase 2, applying the complementary rules, A and T and G and C. Then I will be generating a complementary DNA, which is not existing in nature, right? Because it's random. Now, how do you ensure it does not exist in nature? That's where I'm applying synthetic engineering. And I'm including a new base. I named it as N in this particular system. So instead of AGCT, any one of the base can be replaced with N. So that's the phase 3 part. And then I'm going with phase 4. As you can see, synthetic DNA, there are two different types of mapping I had used. That is the mathematical, that is using gray coding to map DNA with respect to the 00, 01, and then 10 and 11. These are the 4 possible value one of the base can have. I'll show the table later. Say A, if A has 00, all the possible other values also A can have. And then depending on how I am mapping, that is the first stage. And second mapping includes mapping that integer values. I had randomly taken some 0, 1, 2, 3, 4 values. Depending on your requirement, you can have a database accordingly. Then after mapping, what I am generating is a unique code. So here, till here, the encryption, the process encoding is completely done because there is no key involved here. So whatever I am generating, that's the major part I have to look into for security. That is the synthetic engineering part. Plus now I have a unique code. Now how it can be used, I'm going to use a QR code generation because MHOFW says using QR code generation is advisable. So that's how the usage of QR code comes into picture. If anybody of you have a QR code scanner, you can scan and see all the values has been embedded. Like the proposed system, I had done a proof of concept. So whatever I have done, I had actually embedded that values in this particular system, which has not connected with a hyperlink, but you can see the values right now. So as a user, what I'll do when the product I receive, I will be having a scanner like this QR code in this one. So I use a scanner, which will give me the code. So when we are going live with the products, once it is implemented in real time with the products, what we are planning is there will be a link where user can enter the code and then check whether the product is genuine or not with respect to authentication server. As a user, what I want to know is whether the product is genuine or not, which I'll be getting a message from the authentication server, which the system is connected to. Because there will be a connection, whatever the pharma companies they are providing, there will be having a database connectivity. So where they will be showing the product is genuine or not. This unique code will be with them. So as a user, I got the product genuine or not. So as a user, it ensures usability to me. Plus, it is also telling the product is ensuring authenticity. The product is genuine because it has been checked from the database and the server has verified this data is already present there and the product is genuine. So this is the proposed system for drugs or maybe any product we can consider. Now this is a small POC part. I had included two to three screenshots. This has been implemented using Gene Designer and MATLAB. MATLAB does supports bioinformatics tools where we have a sequence tool. That is what I had used to implement this. As you can see, this is the sequence which I was referring to. So all the bases, as you can see, the first stage of importing the gene sequence is what I had generated using Gene Designer. You can see the percentage, the base count here, what I have used for this, and the percentage used as a base count here. That's the first part. Then as you can see, there are features available like reverse complement sequence. What I have done is generating a complement sequence. If you look at the image, you can see here CGT are the only three bases it is listing here. In the previous one, it's all TTCC, GT, GAGA. If you come here, you can see instead of T, there is A, and instead of C, what comes next is G. So basically A to T and then C to G. That's what the complementary rules which I was referring to. You can also design your rules since we are going with the synthetic DNA part. Depending on the synthetic engineering technique, what you are implementing, you can go with the rules accordingly. You can define your own complementary rule, though the base rule, what we follow is what's the complementary rule. So once this is done, next part is the mapping, which I had informed. There is a proposed system will be having a mapping. That's what I mentioned, right? So what I had taken here is N as the base. That is the synthetic engineering part. This is the base which I had included. Only one base is what included. As you can see, N, A, and GC. So one base has been replaced with a new base. That's the reason this is known as synthetic DNA. And then comes the gray coding part. This is what I was referring to. This is the default gray code which normally we follow when we go for DNA cryptography technique. If we have to follow a gray coding part. This is the first part. And then mapping, as you can see, this is randomly taken for integer 1, 2, 3, 4. If any kind of mapping can be done for randomness, maybe you can also generate random numbers in this case. Depending on the high-end security, what you required for the product, then you can map with this. So once this is mapping is done, what you generate is the Unicode, which, as I mentioned, is embedded in the QR code. You can see in the previous QR code, I had not implemented this, kept it again because the same code is what I had kept in the proposed system. So once this is done, the unique code will go as the labeling part you can use. So this is actually a kind of labeling technique which has been proposed using synthetic DNA. So I hope the synthetic DNA part is clear. Now, DNA cryptography is also something which I had discussed, and this is the analysis part which I am showing the complexity. As I mentioned, reverse engineering is very difficult. This equation shows that. How do I claim what I had proposed is a proper one? So that's where the formal analysis of the protocol has been done using the tool CIDR, and you can see all the parameters, whatever I had proposed has been verified, and okay, all the orders and the parameter exchange has been verified. So that's where NIAGREE and NISync comes into picture. Now, what is the PKI connect here? So you might be thinking, I had proposed the system, right? So RFID and PKI is something already existing. Now the plan is to come up with what I had proposed is with connecting with PKI so that I can also include right now only product to user. This is what's happening. So the PKI connects right now, it's working on the supply chain as well. So where we can implement using PKI. Now, that's the conclusion of whatever I had discussed so far. So proposed anti-counterfeiting method, the method what I had proposed is novel in terms of the synthetic DNA keyword, and the POC and analysis part is done for the protocol. And these are few of the references. There are many, I included only 2, and yeah.
Thank you, ma'am Sreeja. Thank you.
Thank you, sir. Thank you.